I did start my journey in this particular field when I was at US doing my postdoctorate. There, it was the time around which CRISPR technology was discovered, 2012, 13. And I had the privilege to work with the best researchers, best scientists in the field in US. And now, since the last 10, 12 years, this field has seen an exponential growth. More so because 2020 Nobel Prize was given in this technology as well. What we see currently is a lot of disease are being treated using CRISPR-Cas technology. So the application of this particular technology are endless. And what we have seen over these last 12 years, there is, besides CRISPR-Cas, there is a growth in the similar genome editing techniques. And there are modified versions of CRISPR-Cas now. We have base editors, we have prime editors, and they have equally seen, even in some cases, they have shown much better genome editing efficiency, as well as when we see the application of these techniques. So they, particularly base editors, have been shown to be much more sophisticated with less off targets when we talk about using them as therapeutics for curing human disease. So this is the recent news. December product using CRISPR-Cas9. This is the conventional Cas9. So I'll discuss in this presentation what do we mean by conventional traditional CRISPR-Cas9 and what are the evolved versions of this Cas system. So CRISPR-Cas9, conventional CRISPR-Cas9 was used to develop this technology, which is a permanent cure for sickle cell and beta thalassemia. So then this product is named as Casgevy, and the approval by FDA as well as by European authorities was obtained last year in December. So I'll be happy, sir, if in between there are any questions, students can ask me, and they are welcome to interrupt me and ask. If there is any doubt, we can also discuss that. So what we have seen is this is a recent news where Casgevy is
Casgevy from Vertex Pharmaceuticals and CRISPR Therapeutics, and that's the first gene editing treatment approved here in the US.
So what does that say is these techniques and technologies are not just limited to the labs, and now they have seen an unprecedented application for treating human disease, besides being used for many other applications. So now Cas-JV is the first genome editing cell-based therapy, which is approved for beta cell and sickle. And the publication was in 2020. Two back-to-back publications came in 2020. One is which is shown here. It was published in NEJM. And what do they target in Cas-JV is this BCL11A, which is a regulator hemoglobin gene. So this is the gene which has been edited. The regulatory region of this gene has been edited, and the technique using targeting BCL11A has been successful, because now we have more than 100 patients who have been treated with Cas-JV using CRISPR-Cas9. So genetic engineering is not something new. We have been doing modifying organisms almost from last 50 years now. And why is CRISPR-Cas so prominent in terms of doing these genome edits? Prominent as well as why do we have seen such an unprecedented as well others. For example, we gene mutation, the typical example we have in the normal, all of us have a GAG sequence, which makes the normal hemoglobin protein. This gene is located on chromosome 11. Sickle patients, there is a single base substitution where A is converted to T. So GAG, so in sickle cell, you have GTG, just a single mutation, and then that single mutation converts glutamine to valine, and it is because of this single based mutation we see unhealthy RBCs which are being formed because of the low levels of hemoglobin gene. And currently, base editing is being used to correct this single nucleotide change, and CRISPR has been used to do change at the other regulatory regions. But even with the CRISPR, we have not been able to do a single gene correction. But thankfully, we have now this base editing technique where we can a single gene correction can be done. And this sickle cell, we have a couple of publications also from different groups across the globe where they have been successfully used base editing to make a single nucleotide change. And then from normal, from mutant protein, they have been able to correct to the normal. And as I was saying, it is not just a new technique. We have been doing genome editing since almost 50 years now. And since the 1990s, we have zinc finger nucleases, which were used. And then came talins. Talins were discovered around 98, 99. And then recent addition is CRISPR-Cas technology. So zinc finger and talons, they are also equally good, but the amount of time these techniques need to edit the genome is humongous. That is the reason people have switched to CRISPR-Cas. And it's not just limited to the cell single gene corrections. With CRISPR, people have been able to do the corrections in the whole embryos, in the human embryos. And then there was a study in 2018 published in China where the CR5, which is a receptor for HIV, was deleted. And then those children who were born from the in vitro fertilization edited embryos, they are as of now healthy, and they are probably they will be HIV free if even if they get the viruses. So that gene has been deleted in those. There are ethical considerations of how and where we can use this technology, because this technology has to be used for to alleviate the suffering of the human beings rather than creating the designed babies. So there is a another, like that is another a debate and a topic of interest where we have now clear guidelines which conditions and where to use this CRISPR-Cas technology. And then there are genetically modified foods. Mushrooms have been used, made using CRISPR-Cas technology. So their applications are endless, and that is the reason these two ladies, Emily Charpentier and Jennifer Doudna, were awarded the Nobel Prize in 2020 for this CRISPR-Cas technology. Okay, so when we look at the timeline, so when do we see what was the origin of this CRISPR-Cas technology? It is not something new. The original discovery of these CRISPR loci were made by Yoshizumi Ishino in 1987, working in Japan. So he made the first discovery that there are repeats present in the prokaryotic cells. So this tool and technology is derived from the prokaryotes, bacterial. The origin is bacterial. And then in 2002, the first genome sequencing of the prokaryotes revealed that there are these clustered, regularly interspaced regions present in most of the prokaryotic systems. And then in 2005, uh, these repeats were found to be from the not from the bacterial origin. They were found to be from a different species. For example, they were found to be originated from the viruses. So those repeats are derived from the viruses. In next couple of slides, I'll tell you how those repeats are derived and what do we mean by CRISPR locus. So just a brief overview of the timeline. So 1987 was the first time when we discovered there are these regularly interspaced regions present in the prokaryotes, bacteria. And then 2020 was the time when Nobel Prize was awarded. And since then, this technology has seen immense growth and potential for endless applications, both across the organisms. 2017, we saw that CRISPR-Cas technology can also be used for molecular diagnostics, like we have PCR, real-time PCR. And 2018 to 2020, we have seen the next generation CRISPR-Cas technology, which are base editors and prime editors. Okay, so what do we mean by CRISPR? So CRISPR, that C stands for clustered, R regularly, I is interspaced, S is short, P is palindromic, and R is repeats. So that is how we abbreviate this CRISPR. So CRISPR is a locus which is present across prokaryotic system. And what, as you can see in this particular schema here, so CRISPR locus is present in the genome of the bacteria, and this locus consists of repeats and spacer regions. So these repeats are called as direct repeats, which you can see as a square here, and then these spacers which are in between. So repeats are in the denoted in the by black squares, and then spacers. So these spacers are the sequences which are derived from the viruses. So this system was discovered in bacteria, similar to the system that we have in humans as an immune system. So bacteria derives these sequences from the viruses, so that when there is a subsequent infection by the viruses, bacteria are ready to fight against that viruses by transcribing these sequences, so that these sequences will go and bind to the virus and kill the viral DNA in the process. So how it works is, virus, when it infects the bacteria, it releases its DNA, which is shown in this particular example, although virus can also insert RNA into the bacteria. So when this DNA is injected into the bacteria, this DNA is cleaved by a set of enzymes, which are Cas enzymes present in the, those are different, like Cas1 and Cas2, present in the bacteria. These enzymes cleave these foreign DNA from the virus and makes it in short repeats of 20 nucleotides. And those 20 nucleotide sequences are then incorporated between the direct repeats. So, for example, this red sequence. So this way, multiple of these sequences are derived from the viruses, and a virus is made ineffective. And later on, when second time this virus infects the bacteria, these sequences are transcribed along with the direct repeats, and these transcribed sequences then get matured. So there are three processes: acquisition of the foreign DNA from the foreign DNAs from the viruses. Second is CRISPR RNA processing. So once this DNA CRISPR locus is transcribed, it makes the pre-CRISPR RNA. This pre-CRISPR RNA is then further processed inside the nucleus, which generates the final mature CRISPR RNA. So whenever these viruses are infecting the bacteria a second time, because the origin of these spacer sequences is similar to the virus DNA, they will be complement. So now in this case, it is the RNA which is made by the bacteria. So this RNA will be complementary to the DNA of the viruses, and whenever the bacteria sees these viruses, these mature CRISPR RNA will go and bind to the DNA of the virus, and that in turn will lead to the cleavage. So what happens during this process between stage second and stage third? This mature CRISPR RNAs are then associated with an enzyme which makes a functional CRISPR-Cas complex. The enzyme in this case is Cas9. So this functional CRISPR-Cas9 then together binds with the foreign DNA from the viruses. And in turn, once it finds the complementarity with the DNA of the virus, this Cas enzyme gets activated and leads to the cleavage of the infected virus. And this is how we know about the system, and which has been existing from millions of years. And it was in 2012 when Jennifer Doudna, Emily Charpentier, and Fang Zhang, they studied this system, and they found that this system can have endless application in doing precise genome editing. So this to make it a little more detailed, the pathway, there are these three steps are further divided into seven sub processes. So first is DNA invasion, that is the viral DNA gets invaded into the cell, and then invading DNA is incorporated into CRISPR locus or CRISPR array, and then pre-CRISPR RNA is made from the DNA, and subsequently guide RNA is made, and then there is this important component which is called as tracker RNA or trans-activating CRISPR RNA. So this trans-activating CRISPR RNA falls is upstream of the CRISPR locus, and it is always present there in the bacterial system. So when this guide RNA, this pre-CRISPR RNA is transcribed from the bacterial system, this activating RNA is also at the same time transcribed, and they together combine because of the complementarity in their three prime regions to make a functional guide RNA. And this is what is now called as single guide RNA. Single guide RNA, which has a tracker RNA as well as a guide RNA. So what it does is this tracker RNA recruits the Cas enzyme, and the guide RNA binds to the complementary invading DNA from the virus. And this final CRISPR-Cas complex is made, and then it leads to the cleavage of the viral DNA. And that is how this system works in these 7 steps. And in 2012, Jennifer Doudna, Emily Charpentier, and then Fang Zhang, working at the same time at MIT, he all these groups together. Jennifer Doudna and Emily Charpentier were working together, and Fang Zhang was working separately. But both the groups together, they found that this system can be made simple, and if we want to make change in any of these cells, we can utilize this system, engineer the system to do the precise genome editings. So what we need in this from the bacteria are two essential components. One is single guide RNA. So there are actually 3 steps in the whole process after engineering. One is the guide RNA, then we have a CRISPR-Cas9 enzyme, and the target DNA. So Cas9 enzyme is derived from the bacterial system. Guide RNA is also part of the bacterial system, but now depending on what we want to target, we can synthetically design this guide RNA. And then the third component in this CRISPR-Cas tool and technology is the DNA, which is the target DNA which we have to target. So Cas enzymes can be Cas9, which is the conventional and traditional Cas enzyme discovered. Cas12 and Cas13, they are also the subsequent versions of CRISPR-Cas system. Cas12 has been shown to have a much better editing efficiency, although it also has higher chance of off-target effects, which I will discuss in subsequent slides. And Cas13 is the enzyme which makes edits in the RNA rather than DNA. So Cas9 and Cas12 makes edits in the DNA itself, but Cas13 is the enzyme which makes edits in the RNA. And then we have single guide RNA, sgRNA, which is composed of, as I mentioned earlier, tracker RNA as well as the CRISPR RNA sequence or the spacer region. And then the last component is the target DNA. Target DNA in case of Cas912 and target RNA in case of Cas13. Cas enzymes. So Cas9 has a peculiar feature. I'll briefly touch upon this. It has two domains. One's called as HNH domain, and second is called as RUBC domain. So these are the domains of Cas9 enzyme, which after binding to the target DNA sequence causes double-strand DNA breaks. And these enzymes have been further engineered to create either single-strand breaks, which is called as nickase, Cas9 nickase, or the enzyme which does not lead to any genome cuts, that is called as dead Cas9, where both HNH domain and RUBC domain have been mutated. So those are the evolved engineered versions of this Cas9. And the important part in this Cas enzyme is this PAM-PI domain. That is a PAM sequence which is present in the target DNA. So we can edit any genomic sequences provided that genomic sequence has this PAM region. If this PAM region is not present, then this conventional normal Cas9 cannot act. So the limitation of this system is that the enzyme depends on a PAM sequence present in the target domain. So PAM sequence is N, which stands for any of these 4 nucleotides, and GGG. I'll show in the next slide. And single guide RNA is the one which we use to target our gene of interest, and then the target DNA can be anything. So this is how this PAM sequence looks like. PI domain of the enzyme binds to this sequence in the target DNA and can be any of these 4 nucleotides, and then it has to be GG. Although it has been shown that after every 8 nucleotides, we have a GG sequence in the human genome. But that is also a limitation that if you want to do a precise genome editing, because of the limitation of this PAM sequence, we cannot do so. Now we have evolved versions of these Cas enzymes, which are called as PAM-less Cas enzymes, where the PAM is not needed. And as was mentioning, single guide RNA. So guide RNA has two major components, and this is what Jennifer Doudna and Fang Zhang eventually discovered. So one is tracker RNA, which is around 65 base pairs, and then second is CRISPR RNA, or it is also called as a spacer sequence, which is around 36 base pairs. So together they are made as a single RNA sequence or RNA molecule that is called as single guide RNA. So overview of genome editing, we have Cas9 enzyme. This Cas9 enzyme is available almost in like 50% of the labs across the world. And then we have a guide RNA. The guide RNA is the one RNA which where you design this spacer sequence based on the gene of interest that you want to target. And then we have guide RNA plus Cas enzyme together forming the Cas9 gRNA complex. And once this Cas9 gRNA is introduced into the cells, it will go and specifically find the target DNA and then lead to the cleavage of the target DNA. And that way we can create a genome edits. Both knock-ins and knockouts can be generated this way. And if it is a knock-in, then NHEJ pathway, we need to have a donor DNA. And so now if it's a knockout, then we have NHEJ pathway, which leads to insertions or deletions. Those are called as indels. And if it is a knock-in, then the pathway will be a double-stranded break repair, where we need to provide a donor DNA. So
the CRISPR-Cas9
this
is a
system
tool for cutting DNA.
This is a animation of how this system works, and it was published in Nature, where you have the DNA, and then the Cas enzyme and guide RNA will go and find these specific regions. So this is our Cas enzyme with the guide RNA complex. Remember, this is the tracker region of the guide RNA, which complexes with the Cas enzyme, and its spacer region is open to make a complementary reaction with the target DNA. So once this system is introduced into the cells, it goes and scans the DNA, and wherever this guide RNA finds the exact complementary sequence in the DNA, it stays there, and then this Cas enzyme will get activated. Both the domains which I have shown you, they will cut. One domain will cut the upper strand, the other will cut the lower strand to create a double-stranded break in the genome. And once this double-stranded break is created, then we have the repair pathway, which will lead to indel formation. And this way, we can create genome gene knockouts as well as knock-ins. Similarly, this technique has been used to also regulate the expression of the genes. It is not just making genome edits, but it can also regulate the expression of the genes. In that particular example, we have Cas enzymes which are causing nicases. So those nicases can also do single nucleotide change, provided we have a specific PAM sequence on the site. And then regulatory regions, regulatory elements have been added to the Cas enzyme, where we have made a dead Cas enzyme. So this dead Cas does not cut the DNA, but it stays at a particular region in the DNA, and then it can lead to either upregulation or downregulation of the genes. So those are the regulatory Cas enzymes. So coming to the sgRNA design. So guide RNAs, which are the essential part of Cas enzyme, we can get from any lab, and it is available at different sites. AdGene also has a repository for Cas enzymes. Guide RNA is the major component which you, whenever you are performing your CRISPR-Cas reactions, you need to design your own guide RNAs. So guide RNAs can be designed by this tool. So here is a brief overview of how you can design your guidance. You can go to Google and to ChopChop. ChopChop is the site which was developed by MIT. And then in the ChopChop, you can type, this is your target gene which you want to target. For example, we have P53 gene to be targeted, where we need edits. And this Homo sapien, we can do it for all the possible species. And then we have on the other side, we will be having CRISPR-Cas9. This is the enzyme which we'll be using. Nickase, and then CPF1 is the Cas12, and then Cas13 or talents. And then if we want to generate knockout or knock-in, so we have to feed all this information, then go find targets. There it will come up with a list of all the possible guide RNAs for your gene of interest, and the location, genomic location of those guide RNAs will be mentioned, and the length at which this genome editing will be taking place. And these guide RNAs will be ranked based on they should have minimal off-target activity. So these guide RNAs should not bind to any other sequence in the genome. They should only bind to your gene of interest. So that is MM01 to MM3 means the amount of off targets they have. So the first guide RNA will be always the best because it will only bind to your gene of interest and it will not bind to any other sequence. So you choose first guide RNA, and then you click on that first guide RNA. This chop tool will allow you to take this particular sequence of your guide RNA. See, GG is the PAM sequence, NGG present in your target DNA. And you can copy this sequence, and then you can use this sequence to order further. And then you also have enzyme primers for genome sequencing of this. When you perform the genome editing, this particular tool also gives you the primers so that you can subsequently check whether the genome editing has taken place or not by PCR as well as by sequencing methods. So this is the important tool, ChopChop, which you guys can use if you want to design novel guide RNAs for your experiment purpose. And generation of the gene knockout and knock-ins is another application of this technology. So there are limitless applications, as I mentioned. We can create gene knockouts in mice, in cells, as well as what we see recently. There has been a Cas enzyme along with the lipid nanoparticles have been introduced into the humans also to treat some disease. So in terms of our genome editing based knock-ins and knockouts. So this technique is simple. We have this Cas enzyme, then we have target DNA. So it leads to the cleavage of the double-stranded DNA. And once the double-stranded break is created, we have NHEJ pathway which comes into play. And then through this NHEJ, we introduce insertions or deletions into that particular sequence. And that is the process by which we create knockout of a particular gene inside the cell. And then we do screening of these cells. There are different methods of checking the exact location where this genome edit has taken place and whether it is a homozygous or heterozygous edit that we have taken place. So this whole CRISPR-Cas system is made simple in form of a plasmid. The plasmid has guide RNA, which you can always change based on your requirement, and then it has Cas enzyme. Cas enzyme will always stay the same. Cas enzyme is driven by eukaryotic promoter, which is Pol2, and guide RNAs are driven by the Pol3 promoters. And this plasmid is also available in most of the labs, and which make this tool so elegant and simple. CRISPR-Cas has been also used in molecular diagnostics. Fang Zhang's lab at MIT was the first to develop a technology which is called as Sherlock. And Sherlock technique uses Cas13. As I was mentioning, Cas13 is different from Cas12 because it uses RNA as a target molecule. And this is how similarly target is RNA, and then we have Cas enzyme along with the guide RNA. It forms a complex. This CRISPR-Cas complex goes to the RNA and then subsequently leads to the cleavage of the guide RNA. And these guide RNAs have been modified along with the reporters to generate the molecular diagnostics. And now we have CRISPR-Cas enzyme available in a simple format, which can be also used for diagnostics in the form of a paper strip. And one recent application of this technology has been for the detection of SARS-CoV-2. As Cas13 detects RNA and SARS-CoV-2 is an RNA viruses, this technique has been shown to work well to detect SARS-CoV-2. And we have our lab has also developed this technique where we have used human saliva sample to detect SARS-CoV-2 in the saliva of the patients. And this is what we have developed. And this is the process of, it's a simple three reaction tube where you can perform the whole SARS-CoV-2 detection. No high-end instruments, no RNA extraction steps are needed. And then this technology was awarded by different funding agencies in India, and our students also got best poster, best oral presentation awards for this developing this technology. And the technology was called as CASPIT, and eventually we made this simple along with a mobile application. So this simple mobile application can be used to detect the whether the patient has SARS-CoV-2 or not. And here is the simple method. You can use this at home for detecting the SARS-CoV-2. And this technology was developed by Iqbal Azmi and Imam Faizan in our lab. This paper was published, patent has been filed. So now what we can see is there are endless applications of CRISPR-Cas. It is not just human genome editing or human cell editing or creating knock-ins or knockouts, but also in molecular diagnostics. And from last 3-4 years, we have advanced versions of CRISPR-Cas. This is the enzyme which is used for first time to treat the human disease. And then these this technology was developed by Iqbal Azmi and Imam Faizan in our lab. This paper was published, patent has been filed. And So I think most of the students have joined. Shall we continue?
Yeah, I think you can proceed, perhaps.
So, as I was mentioning, there are endless applications of this CRISPR-Cas technology. We have whether it is fighting infectious diseases or detecting infectious disease. We have also genome based editing processes where we use this CRISPR-Cas technology to treat the previously difficult the disease which were very difficult to treat earlier, and that part is covered as gene therapies using CRISPR-Cas. And then we also have so with CRISPR-Cas technology, we can also create a comprehensive disease database where we can understand a particular molecular processes. This technology was also developed in 2014, where you use library screening of CRISPR-Cas9. So all the possible genes, for example, functional genes, which we have 20,000, you can do edits in all these 20,000 genes one by one, and then look at how these genes behave in a particular condition. And then we also have people have created almost all the possible cell lines using CRISPR-Cas technology. So in these last 10 years, we have created so many of these knockouts and knock-ins in the cells, which we have not been able to do in past 50 years. And that is the robustness of this particular technology. We also have now CRISPR-Cas based tools where we are trying to make change in the disease, for example, Alzheimer's, Huntington's disease, which have been really difficult. Being a complex disease, we are not been able to find a long-term or a better cure for these disease. And with CRISPR-Cas in hand, we hope we will eventually make it possible to treat these diseases with a much longer duration of efficacy. And then as I was mentioning, CRISPR-Cas for sickle and beta thal is now available. Only limitation with the technology is it is costly. The cost per patient is around 18 crores, so which is not affordable for a common man. So now the other challenge with the technology is how to make this simple technology cost effective. And that is where we are also working, and our colleagues here in India are also trying to make this technology cost effective. And CAR-T is another important application of this therapy. Our group is actively working on CAR-T cells with CRISPR-Cas9, and people have been able to make allogenic CAR-T cells, which are the CAR-T therapy has revolutionized the B-cell lymphoma and leukemia. And now the challenge is again the same cost. And now with CRISPR-Cas technology, we have allogenic CARs where you can make cells from one patient and a normal healthy individual, and those healthy individual T cells can be given to the cancer patient by making these couple of edits in those T cells, which is TCR beta 2M and in some instances PD1. So those genome edited T cells are known as allogenic T cells, or when you incorporate CAR chimeric antigen receptor in them, they become allogenic CAR T cells. And many clinical trials have been done, and the results are promising so far. So that is the power of combining two revolutionary technologies. One is CRISPR-Cas, and another is cancer immunotherapy. Both these technologies have been awarded Nobel Prize. And then first human trial of CRISPR cell therapy, as saying, this is an old article 2020, and now we have in 2023, we have this technology or this product approved for the use for patients. And then we have, as I mentioned, there are endless applications in pharmaceutical. We create a new generation of mice models, rat models, or other animal models to study a particular phenotype or to study a particular disease. We also can create different varieties of plant-based products, and also disease-resistant crops have been created using CRISPR-Cas technology. Biofuels is another application. Targeted medicines, as I was mentioning, is another important application. While doing all these changes with this revolutionary technology, one has to consider these ethical concerns. So this technology is meant to benefit the human society, not to create a harm. While there are people across globe who have used this technology to create designer babies, which again has a huge ethical concern, and then making superhumans, that is also a big concern. And normally those edits and those changes are not allowed. So only application as of now for this CRISPR-Cas technology is to make humans as for the disease treatment, or to enhance the productivity of the crops, or to make animals also disease resistant, crops disease resistant, but rather than enhancing the naturally existing features of an organ or organism. Although that is possible, but again, these are considered unethical. So there have been transgenic animals which have been shown to have much better editing efficiency and much better phenotype than the transgenic animals which we have created using other technologies. And then future of CRISPR diagnostics is also bright. Jennifer Doudna, she has been actually working on the Nobel laureate. She has created a couple of really amazing technologies which have shown robustness in detecting viral and bacterial pathogens. And those techniques are, as I was mentioning, similarly integrated with the smartphone and other devices. And then besides CRISPR-Cas9 normal techniques, we have super precise CRISPR tools, which is an enzyme recently evolved to show much better efficiency and low off-target activity. And then, as I was saying, nucleases, CRISPR-Cas. Now, from 2020, we have base editors and prime editors. Base editors or prime editors have similarly revolutionized this CRISPR-Cas field, because here there are single nucleotide changes which you make, and those nucleotide changes do not bring any off-target activity. And the prime editors are also amazing tools where you can make knock-ins without creating any significant deletions in the genome. So these are the two recent evolved techniques of CRISPR-Cas platform. And as you can see in this particular video, this is a recent last year report where the CRISPR-Cas technology was used, as I was mentioning, to edit T cells of a patient. And those T cells were then given back to the patient who was having blood cell leukemia.
Leukemia, you hear all the good things. She's got the most common type of leukemia, like 96% of children are cured, and it's just very hard when you're that 4%.
It turned out that chemotherapy drugs she'd received weren't enough to control the disease. In fact, she'd already had a bone marrow transplant previously, and unfortunately, the disease had still come back. She was running out of treatment options, so we're looking at trying to make a new treatment for her. We've taken immune cells called T cells from a healthy donor, brought them to our laboratory, and we've changed them so they can work against types of leukemia that are otherwise difficult to treat.
I'm feeling happy, actually.
Why are you feeling happy?
Because I have had new cells put into me.
Really?
Yes. Wow.
Because Alyssa's leukemia hadn't responded so well to the chemotherapy and was quite aggressive. They said, really, really tired,
So like Alyssa, we have so many other patients, kids particularly, who have been recently given this CRISPR-edited CAR T cells, and the results have been impressive so far. So that is the endless application of this CRISPR-Cas technology. And with this, I will acknowledge my lab members who have been actively working and helping me to develop more understanding about CRISPR-Cas technology, and my collaborators, Dr. Shiva Prakash from IGIB and Dr. Gaurav Kharia. He is a senior consultant and bone marrow transplant surgeon at Apollo Hospitals, and we work closely to make this CRISPR-Cas technology affordable and to reach to the patients who are in need. So as a clinician, he is always there helping, supportive. Dr. Shiva is working again in sickle cell area, CRISPR-Cas. He has worked with a person who discovered zinc talins, the prior genome editing technology, and then he also worked on CRISPR-Cas, and then all the lab members who have been actively working. And I'll be happy to take questions and have a discussion if there are. I think we have good amount of time now, and I'll be Happy to take the questions.
Uh, Thank
Thank
you
you
very
so
much,
much, sir.
Dr. Tanveer, for a very comprehensive and very nice talk. I think though it's a very basic, because our audience are very preliminary background knowledge students of BSc and MSc students. So I think for
Right,
them
sir.
the was very good. They can learn a lot on it. And if any question from the audience, please, we have a couple of minutes, 5 minutes at least, to discuss Dr. Tanveer. Please, Apsar, if you can take questions from chat box or from directly, because there are not many. Yeah, please.
So it can be also general discussion if some of these students want to know about the future
Yeah.
of this technology. If they are interested, they can also come to our labs and learn here with the people who have been actively working on these techniques. So it can be also a general discussion, sir, I think.
Good afternoon, sir.
Yes, Safana, good afternoon.
I want to ask that can cancer be treated by CRISPR-Cas9 by removing genes or correcting mutations that are responsible for mitosis? And what are the
Uh,
challenges?
Excellent question, Safana. That is a much bigger challenge. So what we do is with CRISPR, we can do single genome corrections, editors. That is why this technology has been successful in treating the disease which are inherited disease, genetic disease, and genetic disease with mutations or deletions, like like monogenetic disorders where you do not see a complex kind of gene disruptions. That is the reason CFTR, sickle, thal have been successfully treated with CRISPR. ATTR is another, hypercholesteremia genes have been another success for CRISPR-Cas. But for cancer, because it is not a single gene disease, and what we know now, cancer is associated with mutations and changes in multiples of those genes. So as of now, only CRISPR-Cas alone has not been successful to treat cancers. But now what we have done, and other group across the globe, they have done working in the field of immunotherapy. So we use CRISPR-Cas and then we use immunotherapy like CAR T cells. So we combine those two techniques and then we take out the cells, these engineered cells, and we take the cells from the patient, introduce CRISPR-Cas system into those cells, make the edits, and then incorporate a receptor which is called as CAR, as mentioning, and then we introduce these cells back to the patient, the person who has cancer. Even stage 3, stage 4 cancers have been treated. And though this is how CRISPR-Cas has been successful in treating cancer by combining with another approach like immunotherapy. Alone, as of now, we are not being able to see success in case of cancer because of the complexity of the disease. Is that clear, Safana?
Yes, sir. Thank you.
Okay, very nice. Anyone else, if there are any other questions? So, sir, if we don't have any further questions, shall we then conclude the session? Hello,
Yeah, I think you
Absar,
can.
can you hear me? Okay. Mhm. Absolutely true. Yes. So, although Alzheimer is also a complex disease, but because there is a protein involved which is called as APP. So, there have been groups who have been working to edit APP. So APP editing has been shown to do not cause any side effects. So even if this gene is deleted, it does not cause any change in the neurons. And then people have tried it in model organisms, but so far in humans it has not been done. But in model organisms it has been done. By disrupting this APP gene, the uh, pathogenesis and the progression of Alzheimer's have been significantly reduced. And for humans, it has to be yet to be seen how well we can do it. But yes, there is a possibility that in future we can see CRISPR-Cas treating Alzheimer's disease. So that is another interesting question, actually. So all these complex disease, we have yet to see the success, but all the disease which are associated with single nucleotide change or deletions or insertions, genetic disorders, we have been able to see unprecedented success in utilizing CRISPR-Cas.
Uh,
So,
Tanvir,
yes, sir.
if you allow, I can ask one question.
Yeah, absolutely. Pleasure.
Okay. Basically, my area of research is, is antimicrobial resistance, and major focus on
Right.
biofilm as the virulent factor. So earlier we tried to attempt in vitro that how CRISPR-Cas. So we designed CRISPR-Cas, CRISPR-I basically to knock down
Right.
against several biofilm or quorum sensing genes in E. coli and Pseudomonas. And we successfully in vitro, we have shown that they are completely turned down the gene. Even we have shown in Fimbriae genes that the Fimbriae is completely lost in the transformed with CRISPR-Cas into the E. coli cells. So we've shown through transmission electron microscopy and SEM that all Fimbriae are lost in case of expression of CRISPR-I. And that was published in some immunology, Friend Immunology, and other journals. But we face a lot of comments from the reviewer that how can you treat infection having such kind of a bacterial, having the biofilm forming bacteria? That was a challenging question for me. So what I did it that we have shown them that transformation of the whole cassette CRISPR-Cas, which I designed in lab, to in another bacteria. And by transferring them, they remain intact, the character of CRISPR-Cas, and they have shown the same character into the E. coli, as per the phenotypic, that is, reduction in the biofilm is concerned. So keeping that in view, we justified it with this preliminary experiment. We justify the reviewer or the editor is that we transfer the engineered bacteria into the gut, and we try to modify the the genes of resistant genes or virulent genes by horizontal gene transferring from one bacteria into another bacteria into the gut or infection site. So perhaps he convinced and the paper got accepted. But I think still I'm not myself is not convinced with this, because suppose if we have a severe infection and the infection is caused by Pseudomonas, like lung infections, and Pseudomonas is over a period of time, they start forming the biofilm formation.
Right.
So how would we proceed as a CRISPR-Cas CRISPR-Cas molecule are given to that infection site, just like we use antibiotics, so that it can wipe off the biofilms and cleave the infections. This is, I think, to me, is not very acceptable. If you can address that question.
Right. So this is amazing, sir, the approach you have used, because it has been shown by different groups that it is really difficult to use this CRISPR-Cas technology for the editing of the bacteria, because the original source is bacteria itself, right?
Exactly.
And it has been really difficult to edit, do edits in the bacteria. What happens is bacteria does not allow any changes with this technology. And now, sir, there are interesting discoveries recently. This CRISPR-Cas system has been shown to be present in other species other than bacteria. So there is viruses also have these sequences, and then there is yeast, species of yeast, which has also been shown to have this CRISPR-Cas system. So in the approach that you are using, my suggestion is if you can use the CRISPR-Cas system from a different species and then introduce that into the bacteria, so that will allow the edits in the bacteria, because for bacteria it will be the foreign, it will not recognize it as cell system.
Okay.
So that is one part. Second is, which is equally challenging when you are proposing this treatment as an alternate to overcome resistance. So you want to use this as a kind of alternate to the antibiotics. So there the challenge is how you deliver this CRISPR-Cas complex into the bacterial cells. I think that has been the major challenge, because uptake of these molecules into the bacteria is not so easy. So you have to, and then there are cell membrane and cell wall penetrating peptides and molecules which people have used, complex them with the LNPs, and these CRISPR and guide RNAs inside the LNPs. And those approaches people have used, I think, but that is also in the preliminary stage. But this is a
That's
$1 million question. If we can
yeah.
get this done, do something
Anyway, we are
amazing.
trying to do it.
Absolutely,
Okay,
this is
so
a big challenge.
yeah, okay. So I think if there's no further question, I thanks to you and thanks Dr. Tanvi for
Absolutely,
sparing the time.
pleasure,
You are
pleasure,
very
sir.
kind, and maybe in future we are get in touch with you and
Always pleasure, yeah, sir. And I'm really sorry for the technical glitch.
it's okay, no problem. It's all online problems. So
Absolutely.
okay, so now you can
All right.
I think I can start my own WebEx platform for the other lectures.
Absolutely, sir. Hope to
Thank
see
you.
you soon, sir. Bye, guys. Bye, everyone. Bye.